So I left my hotel at about 9 o'clock, saying, I have enough time. And then it's like, oh, the conference is right 15 minutes away. So I walked there and I'm like, wait, this is not, sorry. And then I was like panicking. And anyway, I'm here, so that's good. All right, so as I was mentioning, we had Mo did a really nice deep time course from fibroblasts to iPSCs, reasonably finely sampled time points, but not finely enough, as I'll show you in a few slides. So you overexpress these factors using Sendai virus vectors. You infect the cells, and then they reprogram, or you hope they reprogram. So this is the UMAP of the cells from the ATAC-seq data, and they are colored by the days, as you can see. And you'll immediately see there's really interesting heterogeneity. So we start off with these day 1 cells, and then you have right at day 2 you see quite a dramatic. Don't overinterpret UMAPs, okay? But I'm going to overinterpret them for you now. Here's the fibroblasts. Here's the iPSCs at the end of a pretty long time course. And you can see there's lots of teleportation going on. There's like fibroblasts right here. These are day 1 cells, and then day 2 cells have already diverged, and there's day 3 and day 4, and then things start diverging pretty fast. So you get very rapid cell state transitions, and then finally, after many passages, you end up with these primed iPSCs, okay, from naive iPSCs right here. So instead of focusing on the days right here, we can focus on the standard, like we can cluster cells. Clustering is obviously not a good idea in this sort of situation because there aren't like disjoint clusters. It's a very continuous process. But what we'll do right now is we'll use clustering mostly to get landmark substates of cells. I will not call them cell types or cell states. Just think of them as landmarks. And we're going to try to pseudobulk populations and study them because we currently do not have single cell models, but tomorrow we will. Jacob Schreiber is going to present a great talk on how we have extended some of our models to truly single cell resolution. But for today, I'll focus on pseudobulk style models, okay? And so, we have 15 states. They are, again, approximate. The only 2 real like states in some sense that you can anchor on actual cell types or cell states is fibroblasts right here, iPSCs right here, and then everything else is kind of intermediate states. You have fibroblast-like cells which did not seem to reprogram. You have a weird keratinocyte-like state that emerges in day 2, and the cells terminally stop right there. There's a state called a high OSK. I'll show you why. It's like super high expression of the transcription factors. Then a bunch of intermediate states. Ultimately, you get pre-iPSCs. There's some partially reprogrammed cells that keep falling off the primary trajectory, and then you get iPSCs in the end. Okay, so don't focus too much on what exactly the cell states mean. Think of them just as landmarks to try to capture different cell compositions across the entire time course. This is the RNA and the ATAC, and you can see pretty nice correspondence between them. Suragi used CCA to link the two together, and we pick up pretty much the same populations. Again, you have fibroblasts, iPSCs, keratinocyte-like cells, bunch of intermediate states, pre-iPSCs, and so forth. So if you start drawing arrows, which people love to do with single-cell data, here are some arrows. What we're seeing is we use PAGA here to find trajectories. We also tried RNA velocity, and it gave us completely nonsensical results. So I'm not going to show this to you today. I'm hoping that the newer methods that have come out more recently will work in this setting. But this is one great case study where RNA velocity gives completely very, very strange directions of state transitions. So in this case, we use PAGA to identify these trajectories, and there are 3 or 4 main trajectories. We'll mainly focus on this reprogramming trajectory. How it works is you again have fibroblasts right here at day 0. At day 2, they rapidly teleport to this state. And this teleportation is because we actually aren't sampling fine enough between day 0 and day 2. So it's obviously not a real teleportation, but it just indicates within 2 days there's a massive state switch as you overexpress the factors. And then cells transition along this reprogramming trajectory. And this is at day 14, they're pre-iPSCs. And then there's many more days of passages. Again, we don't have time points all the way to the end, but then there's another teleportation to iPSCs. You can see the There's an off-target trajectory going to keratinocytes. Again, it's a bit of a teleportation. There are fibroblasts that did not reprogram, and there are cells that kind of partially reprogram falling off this trajectory. Okay. Again, I'm not going to focus too much on the trajectories and all that stuff. It's mostly to allow us to get some pseudotime and to order our cell states. All right, so an interesting question is there's tremendous heterogeneity, and then you might ask, why do cells diversify so much, right? Why don't all of them just reprogram to iPSCs? Well, it's actually linked very strongly to the stoichiometric concentrations of OSKM. So what we're going to do here is we're going to paint the UMAP with these state labels, and you see OSKM, and we're going to basically color the expression levels of OSKM in various, so the cells are being colored by the combinatorial expression of OSKM. So in fibroblasts, OXOX, KLF, and MYC are not expressed. So you can see this state where there's very low expression, all the fibroblasts get nicely colored, right? Now let's look at what happens with these fibroblast-like cells. So these are in later days, after the reprogramming factors have been introduced. These clearly do not reprogram because they apparently aren't getting OSKM expression. So since the Sendai virus integration is not deterministic, some of the cells simply don't get the virus. And so when they don't express, they don't reprogram. So let's move ahead. You can see that this keratinocyte-like state is really associated with high expression of KLF and MYC, but not OCT and SOX. So if cells get high expression of KLF and MYC, but not OCT and SOX, they essentially program into a keratinocyte-like state, not to iPSCs. And then similarly, as you move forward, you can see like this high OSK state, this pink one, has supraphysiologically high levels of OXOX, KLF, and MYC. And then these levels sort of start dropping. And then eventually in iPSCs, you get moderate levels of OXOX and MYC. Okay? So what we're seeing is the trans expression of the reprogramming factors pretty much determines cell fate, right? Almost entirely. That makes sense to a large extent. So if you want to increase the efficiency of reprogramming, either you want to increase or reduce the stochasticity of stoichiometric expression of OSKM, and you also want to control the precise expression levels. And people have shown this in many other publications before. Okay, but it's nice to see that it just kind of pops out that way. What we do is, so looking at the trans expression of these factors, we can actually understand how cells are moving to different cell fates. Now let's look at the chromatin accessibility landscape and look at the dynamics of how these regulatory elements are changing. So what Surak did here is, again, now we've ordered the cell states. Now we're going to start looking at pseudo bulk populations, okay? The ones I showed you before, the 15. And we're going to go across this primary reprogramming time course, fibroblasts here, and then iPSCs at the end, and various other cell states across the reprogramming trajectory. And so if you cluster the peaks based on a tax-seq signal, you actually get some clusters that open early and then close. These are open in fibroblasts, and then they shut down in iPSCs. There's a reverse cluster that is closed in fibroblasts, and then they open up in iPSCs. And if you link these to nearby genes, and you look at expression modules, they correspond to exactly what you might expect. Peaks that are open in fibroblasts and closed in iPSCs really enrich for expression of genes that have classic fibroblast functions, like, for example, COL1A, RUNX, FOXL2, FN1, and so forth. The genes in this module, associated with peaks in this module, are classic pluripotency genes, like NANOG, MYC, OTX2, and so forth. But there is a massive population of peaks, and these are really interesting. These are what we call transient peaks. These are peaks that are neither open in fibroblasts, nor are they open in iPSCs. They open transiently, and then they disappear. And you will see waves of them. Some of them open up for a very short time, some of them a little bit longer, some of them a little bit longer, some of them early, some of them late. But these kind of just appear and then disappear. And this accounts for about close to 45 to 50% of the total dynamic peaks. So it's not a small number. It's not like there's 100 peaks. It's like thousands of peaks are just opening up transiently and disappearing. And if you take the genes nearby, again, you see correlated expression of nearby genes, but if you take the genes nearby, and you look for any kind of functional enrichment, you don't find anything. It's like these peaks have no enrichment of any kind of known function. So that's really interesting. So what we try to do next is try to understand what these transient peaks are. There's a huge number of these. The first obvious thing to do is to try to take these peaks and look for them in other like classic known cell types and tissues. So we took ENCODE and roadmap samples, about 2,000 of them. We took DHS data, DNase hypersensitivity data, and we overlapped our transient peaks with peaks in all those cell types. Do we recover them? Do we see them elsewhere? And as you might expect, so the percentage of peaks that are missing, you'll see for the clusters, the fibroblast cluster and the iPSC cluster, most of the peaks are found in other cell types, as you might expect. But if you take these early transient peaks, a massive proportion of them, again, are not found in any cell type. They're clearly open, but they're not found anywhere else. And the same is true also for the late transient peaks. It's a slightly smaller proportion, but a large proportion of them, again, aren't found anywhere else. So it's a strange set of clusters, right? You have these hundreds and thousands of peaks popping up and disappearing, and they're not seen anywhere else, okay, just in the reprogramming system. And if you take other reprogramming data sets, not just our single cell data, but a bunch of bulk data sets, other reprogramming systems, not necessarily using the Sendai virus, the same peaks appear. So it's a reproducible feature of reprogramming, but it's never seen anywhere else. That's really interesting. So we wanted to investigate, how these peaks are being regulated, what's going on. So can we explain these chromatin dynamics using sequence models? Okay. And so for this, we recently developed ChromeBPNet. This is also not yet published, but it's an extension, a pretty major extension of a BPNet model that we published in 2021. It's a base resolution convolutional neural network. What it does is tries to map DNA sequence to base resolution profiles of attack-seq data in this case. You can apply it to DNA-seq data as well, alongside doing implicit enzyme bias correction, okay? I'm not going to spend too much time on this. We presented elsewhere, and the paper will be out soon. I'm mostly going to focus on how we use the models here to study reprogramming. But the architecture of this model is exactly like BPNet. It's the same number of layers. It's a fully convolutional model. It has a profile head and a count head. The count head captures the total number of reads in 1,000 base pair bins across the genome. The profile head captures the base resolution distribution of those reads in the 1,000 base pairs. So there are two outputs. And all I'll say is bias correction, TN5 bias correction, is extremely important for attack-seq. And if you want to know more about that, again, go to Jacob's talk. He'll show you some cool results on that. So we train this model on a subset of chromosomes and then test them on another subset. It does really well across all the pseudobulk clusters. Some of them are, have more cells. Some of them have more reads. Some of them have less reads. This is the count head across 10 folds. You can see, there's pretty nice uniform performance. Some of these models get higher performance. They tend to have more reads. So the performance is correlated with the number of reads in the data set. But they're all in the range of like 0.8 to 0.85 Pearson correlations, okay, in total counts. So that's quite nice. And if you look at the profile correlations, the base resolution profile correspondence, we're using the Jensen-Shannon divergence to compare the observed and predicted profiles. And again, you see pretty nice correspondence. There's an upper bound and lower bound corresponding to replicates versus completely randomized profiles. So performance looks good, models look good. The key point I'm trying to make here is you can apply ChromeBPNet to pseudobulk attack-seq data from single cell experiments, and it works really quite nicely. So we just look at what this looks like. We can take, for example, the lefty gene and look at one of its enhancers, and you can kind of track, chromatin dynamics across all the cell states, and the models track very nicely. The predictions of the models really kind of map very nicely to the observed signal. But the nice thing is now, given you have the model, as I mentioned before, you're using the model to understand how DNA sequence is driving this sort of chromatin dynamics at single base resolution. So we can use, for example, a deep lift, deep shap, which are sequence attribution methods. You can take the model, you can invert it, you can use it to infer base resolution contribution scores for any sequence in any cell state, and you get kind of really nice dynamic sequence maps that look like this. So here you're seeing this lefty enhancer, and you can see, in fibroblasts, there's no OXOX, so this motif is off. There's this Fox motif and an NFI motif that are in this element. And then as you go through the time course, immediately you can see at the high OXOX state, this OXOX motif pops up alongside this KLF motif. The NFIs start disappearing, and the OXOX start taking over, right? So this is very, very nice dynamics. I also want to point out that this is a great example of how the same sequence can be differentially regulated in different cell states. Just because you see peaks, a peak in two cell types, it doesn't mean it's the same sequence elements driving it. You can have different transcription factors going in and out, and these models really allow you to see or visualize and in fact estimate the actual drivers, sequence drivers of that sequence in every cell state. And in Jacob's work, he'll show you that we can actually do that at single cell resolution, in fact. Okay, so that's nice. So what can we do with this? Well, we can take any locus, like NANOG, for example. NANOG is a classic pluripotency gene. When NANOG gets expressed, you start claiming that your cells are starting to show pluripotency signatures. So here we're looking at the NANOG locus. Here's the gene. This is something like a megabase around it or several kilobases around it. And we ordered from top to bottom, fibroblasts to iPSCs across the time course. Okay, these are all the cell states. Here I'm showing the expression of NANOG, and you can see NANOG is initially not expressed, and then it gets expressed pretty late in the time course. But you can see that this enhancer right here pops up very early, quite early, and even before the NANOG promoter actually opens up, right? So the enhancer pops up first, NANOG promoter pops up later, and there's a whole bunch of other peaks that pop up after that. There's also these two peaks that are constitutive. Those happen to be CTCF binding sites, and they are anchoring the TAD. There's a topological domain that NANOG sits in. They're anchoring there. And actually, somewhere in between the time course, another CTCF site pops up. It creates a sub-TAD inside this super TAD, and all the enhancers start playing around with the NANOG promoter and activating it. Okay? There's beautiful chromatin dynamics going on, but the cool thing now is with the models, you can take every peak in every cell state, and you can explain what's going on in the sequence. So for example, here are these CTCF motifs, right, for these two peaks flanking the domain. And then here's the enhancer, right, that turns on. And here's the other intermediate CTCF site that turns on midway through the time course. Here's an NANOG promoter. You can really start tearing apart the sequence logic at single base pair resolution in every cell states. It's quite nice. So for example, you can zoom into this enhancer and say, oh, look what's happening. Initially nothing much, and then a KLF motif pops up, and then a big constellation of OXOX motifs collaborating with KLF and an oct monomer motif. Lots of interesting stuff going on. And you can see also KLF disappears in the final primed iPSCs. KLF expression actually goes away, and the model just learns that the KLF motif is dead. Remember, the model has never seen gene expression. So it doesn't get to see expression levels of any of these factors, but it infers from the profiles, from the footprints of the transcription factors, which motifs are likely occupied and which ones are not. So with this, what we can do is really quite cool. We can take each of our enhancers, we can link them to genes, right, through a correlation or through a distance. We are able to take transcription factors and link them to enhancers, as I showed you before, through the motifs. And so we can start building these kinds of dynamic transcription factor to enhancer to gene networks. So this is a network just for NANOG. Okay, these are these green circles are all enhancers, dynamic enhancers in that NANOG locus. The orange circles are all transcription factor motifs, and you see a link between a motif and an enhancer when it's in that. When you see a light green enhancer, it indicates that enhancer is not accessible. A dark green enhancer is accessible. And a transcription factor that has a bright orange, a dark orange color is an active motif. And if you see a light orange color, it's an inactive motif. So what we've done here is we've placed all the enhancers from all the cell states into one map and the transcription factors. And now you can start seeing transcription factors. Initially, for NANOG at least, you can see the regulatory network is very sparse. The gene is not expressed. And then as you move through time, this is the high OSK state, you can immediately see OXOX motifs pop up. They start occupying several enhancers, collaborating with other transcription factors. Then the network gets really strong. You see lots of collaborative effects across multiple transcription factors, multiple enhancers, and then it kind of subsides away, and you end up with only OXOX and KLF at the end. So you start off with RUNX, AP1, and KLF, and you end up with OXOX and KLF. So you see beautiful dynamics of the network rewiring itself across time. You can also contrast networks of different genes. NANOG is a classic pluripotency gene, so you can see its network sort of activating over time. And here's fibronectin, which is a classic fibroblast gene. You can see it has a super active network at the start, and then that disappears. Right? So you can trace this for any gene of interest and start building these networks quite effectively. Right, so that's individual loci. So let's try to summarize what's happening across the entire genome. So we have another method called TF-Modisco. What TF-Modisco does is it takes these sequence attribution tracks for every enhancer and then finds little subseclets of high importance and then clusters them into non-redundant motifs. And so we can summarize across the entire genome in every cell type or cell state, sorry, not cell type, cell state, cluster, what are the motifs sort of that are active and driving the chromatin landscape. So you can see in fibroblasts, it's really AP1, RUNX, BHLH, sort of dominating the landscape. Immediately at day 2, look at the dramatic transition that happens, right? All of a sudden, this TF lexicon landscape completely changes, and you can see expression levels of OXOX go nuts. They go skyrocket. This is OCT4, this is OX2, this is KLF4. These are OXOX motifs. They completely dominate the landscape. Probably 50% of the dynamic peaks are occupied or show OXOX heterodimer motifs. You see KLF and AP1 starts disappearing really fast. And then over time, you can see the OXOX landscape also starts shrinking, and you end up with stable iPSCs. So what's interesting here is, of course, some transcription factors are appearing and disappearing, but it's more of like this kind of elastic effect, right? Transcription factors are going in, and the pluripotency factors are really occupying the DNA, and then they start disappearing away. And it kind of reminisces, you can link this back to the stoichiometric effect. You have really high concentration of OXOX, KLF, and MYC, and then they start disappearing over time, right? And the chromatin landscape is reflecting that same effect in cis, okay? We're going to contrast these two states because they're both very interesting. They both have expression of OCT4 and SOX2, but the expression levels are very different. So you can see in iPSCs, OXOX and OCT4 and SOX2 are expressed quite heavily, but in this high OSK state where the reprogramming trajectory starts, it's like orders of magnitude higher, right? So it's a completely different ballgame. These are super physiological expression levels never seen in normal cell types, okay? So we just going to contrast the two chromatin landscapes and look at what's different in the OXOX motifs in the high OSK state versus the iPSCs. Are they the same or are they different? So let's start off with the iPSCs. You see the classic OXOX heterodimer. It has 3 parts. It has this piece that binds the POU HD domain of OCT4. This one binds the POU SD domain of SOX2 of OCT4, and then this is the HMG domain of SOX2, the classic OXOX motif. Now, with Modisco, we can actually break down sub-motifs. What are these motifs actually made up of? And then they have slight heterogeneity, but not that much. You can see it's a pretty uniform set. They look pretty similar to the canonical OXOX motif. Now, when you look at the high OXOX state, if you just learn one motif that captures OXOX, it looks reasonably similar to the canonical OXOX motif. But again, we can break down heterogeneity. What is the diversity of motifs that you see in the chromatin landscape? And you see something really interesting. You see a lot more diversity, and some really interesting things start happening. You can see these 3 motifs completely lack the POU HD domain. Like one domain of OCT4 is not being used at all. You have this other motif right here, and you can see that the C in the CAT is replaced by an A. These are really big changes in the sequence. You're losing one primary domain of binding of OCT4. And again, these versions of these motifs are not seen in known databases. So you can't just take a database of motifs and scan the sequence and find these. These are like really non-canonical binding modes of OCT4 and SOX2. And if you look at their actual affinity to the OXOX motif, to the OXOX heterodimer, there's a dramatic shift. So what we are seeing is that in this high OXOX state, OCT4 and SOX2 are binding low affinity binding sites, as you might expect, because the concentration of OXOX levels are insane. They are really, really high. So they can start going and occupying non-canonical binding sites. Whereas in iPSCs, they are sort of at normal levels, and that's when you start seeing the normal motif, right? So this is really a nice advantage of a neural network. It doesn't make any assumptions. You just go in and see what you want to see, and you can pick up all of these beautiful low affinity binding sites, really hard to find otherwise. All right, so let's go back to our transient peaks. I kind of took a bit of a detour, showed you Chrome BPNet and how we can use it to study cis-regulatory dynamics. Let's try to explain what's happening in these transient peaks. Remember, there were hundreds and thousands of peaks, or 80,000 peaks that were popping up and then disappearing. And as I said before, there are at least 3 waves. There's an early wave, very early, turns on and then turns off, this ET1. And then there's ET2 and ET3. And they have different decay rates, right? So some of them turn on really fast. What's really interesting is if we just look at the motifs, the OXOX motifs in these peaks with different decay rates, the affinities almost completely explain their decay rates. So what's happening is the peaks that turn on and turn off really quickly are the lowest affinity sites. So when the concentration of OXOX is really high, they occupy. And then as concentration drops, the earliest affinity sites are let go. And that's the early transient 1 peaks. Early transient 2 peaks have slightly higher affinity. Early transient 3 peaks have slightly higher affinity. And the peaks that stay on across the entire time course, they have the highest affinity. So you can see a really nice interplay between concentration and affinity driving occupancy, right? You have this decay in concentration, and then motifs with different affinities are let go at different times. Right? And that creates this sort of wave of transient peaks. So really quite nicely, we can explain the cis-regulatory control of these transient peaks. So what's happening in transient peaks? They're opening due to OXOX binding weak sites at super high concentrations. These sites are very specific to reprogramming. They activate genes that are transiently expressed that have no real known function in reprogramming. They don't seem to have any conserved activation or any meaningful effect on gene programs. And the sites with the weakest motifs are gradually released by OXOX, leading to waves of transient activity, right? Now, just getting back to this interplay between concentration and affinity, do we see this in footprints? That would be really interesting, right? Because you're having transcription factors binding DNA, and one nice advantage of ATAC-seq or DNA-seq is you should get really nice footprints where transcription factors bind. So here's KLF4. And this system actually is a really nice playground to look at the interaction between affinity and concentration, because as you move across cell states, concentration of these transcription factors are dramatically changing, right? And then in the genome, you also have sequences ranging from super high affinity OXOX to super low affinity OXOX. So with single cell data, you actually have a free playground. Every cell is some sample of concentration and affinity, right? And so we can essentially traverse the entire grid of concentration and affinity, and these are predicted footprints from Chrome BPNet, and these are bias corrected. If I showed you the uncorrected footprints, you would not see any of this. I'm not going to show you that, but Jacob will. Let's start off with the high OXOX state, where the OXOX concentrations are the highest, and here are the fibroblasts, where the OXOX is completely gone, right? There's no OXOX expressed in fibroblasts. So let's just take high affinity sites, and then just traverse, keep the affinity identical and just traverse concentration, and you see this beautiful effect of accessibility going away and the footprints also reducing, right? You can do the opposite. You can keep the concentration of the transcription factors constant, and you can switch affinity. So we can change the affinity of the motif, insert it into the sequence, and you again see this kind of beautiful behavior of as even at super high concentrations, low affinity sites have weaker footprints than high affinity sites. And you can complete the grid. And you see this awesome interplay between the classic interplay between concentration, affinity, and occupancy shown in the footprints. And it's not just for KLF4. You can do it for even transcription factors that people have claimed do not create footprints. So SOX2 is a transcription factor with very low residence time. So lots of papers claim SOX2 does not have a footprint. That is actually true at physiological concentrations of SOX2, but as you go to super high concentrations, you in fact do see a SOX2 footprint, right? And for every transcription factor, we can actually footprint it and look at footprint dynamics as a function of occupancy and affinity. So people who say that like neural networks are useless black boxes, they're not. They actually are sensitive to affinity, concentration. You can look at footprints, you can look at occupancy, lots of really cool stuff. All right, so now the big elephant in the room, what are these transient peaks doing? Right? They're really interesting, we can explain them, but why are there 80,000 transient peaks? What are they exactly doing? So one thing that's happening, I think you may have noticed, is when you switch from from fibroblast state to the high OSK state from day 0 to day 2, really quickly, 48 hours, you must have noticed that the AP1 binding sites disappeared really fast, just as the OXOX sites took over. So there's two things happening, right? You're trying to turn on the iPSC program, and you're trying to turn off the fibroblast program, and both are really important. To reprogram cells very well, you need to do both very effectively. If you just turn the fibroblast program, the pluripotency program on, and you don't turn off the fibroblast program, the cells are going to struggle. They're going to sit in some weird intermediate state. So there's sort of this interplay between the two programs trying to turn on and off. And if you just look at the expression levels of the AP1 factors, so AP1 is the primary transcription factor that drives the chromatin landscape in fibroblasts. You can see really rapid decrease, right? By day 2, AP1 levels are almost gone away. So something is repressing AP1 very, very rapidly. It's not just activating stuff, but something is repressing things very, very rapidly. For AP1, phosphogen, and fibronectin, which is a fibroblast gene regulated by AP1. So you're not only seeing this for the transcription factor itself, but also its targets, right? So there's a really fast repression happening. Now, what's really cool is we can go to ChromeBPNet and ask it, what's happening with these AP1 motifs? What's really interesting is these, these transient peaks that I told you had low OXOX affinity binding sites. They also happen to have AP1 binding sites. So this is weird because AP1 and OXOX are not supposed to kind of be together because OXOX is expressed in iPSCs, AP1 is in fibroblasts. Tens of thousands of enhancers have a syntax of AP1 motifs next to OXOX motifs. And these are not canonical OXOX motifs. These are like these low affinity OXOX motifs. And these are not weird things. They actually have AP1 footprints. So these are definitely bound AP1 sites. These are the transient peaks that turn on really rapidly and then go away, right? Which I just showed you before. So there's a novel syntax we're finding specific to these peaks. It's a syntax involving AP1 and low affinity OXOX sites. So I want to try to understand what's happening here. One hypothesis that people have made is that these OXOX transcription factors, they open up a whole bunch of chromatin, and AP1 binding sites might be, new AP1 binding sites might be introduced. In fact, what we see is 10,000 new AP1 binding sites introduced in 48 hours. These are not present in fibroblasts. They're completely new 10,000 binding sites being introduced as a function of OXOX going and opening these transient peaks with low affinity binding sites. So one hypothesis has been that what might be happening is these pioneer factors go and open chromatin, these transient peaks, they have AP1 binding sites. If you rapidly open up a whole bunch of sites with AP1 motifs, AP1 starts getting relocated from fibroblast enhancers to these 10,000 binding sites. And so you're getting a rapid reduction in accessibility of fibroblast enhancers, thereby repressing fibroblast genes indirectly. Not as a repressor, but by creating a sink. You have a new sponging effect. These transient peaks are like sponging AP1 away from the fibroblast enhancers, and they're turning off the fibroblast program really rapidly. So do we in fact see this? You could maybe test this out using single-cell data. So just to test this hypothesis, Mo actually did a paired multiome, where we took RNA and attack in the same cell, right? Because previously we did not have it in the same cell. So we couldn't exactly tell if RNA levels and chromatin levels are, we couldn't exactly match them up. And so if we do this single-cell multiome, in the early time points, we again recover the same cell states, okay? So it's again the fibroblast-like states, the high OSK and low OSK. So what we can do is, across the single cells, we can correlate the change in the fibroblast program gene expression score and see what happens in different states, cell states. So this is the fibroblast-like states, and we're looking at the z-scores of fibroblast-specific genes. That's what we call the fibroblast program, okay? So it's pretty strong in fibroblasts, as you can see. And the high OSK state has the highest reduction in expression of the fibroblast program, as you might expect, right? So the cell states, or the cells where OXOX and KLF have the highest expression, have the highest repression of the fibroblast genes, okay? What's really nice is, using this sort of strategy, we can actually compute accessibility at newly opened peaks in individual cells. So each point in this plot is an individual cell. Different cells have different levels of how many AP1 sites are opened. So we can get kind of a nice dynamic range of how many new AP1 sites are opened in a cell, and correlate that with the expression level of the fibroblast gene expression program. That's the y-axis. So if you walk across the cells and you just correlate how repressed is the fibroblast gene expression program relative to accessibility at newly opened sites containing AP1, you see this beautiful, really strong negative correlation, right? So there's a really nice correlation between how many new AP1 sites are opened and the repression of fibroblast genes. So we have a refined view of reprogramming dynamics. Basically, what we're seeing is, you have fibroblasts, you have these early states, and then you have iPSCs. The fibroblasts are dominated by let's talk about the trans environment. AP1 transcription factors dominate the trans environment of fibroblasts. OXOX, ZIC transcription factors dominate or determine the landscape for iPSCs. And then these early states, these intermediate states, have super high levels of OSK with all kinds of dynamics of expression. If you take the fibroblast-specific peaks, they're being regulated largely by AP1, as we saw before. What happens then is once you overexpress OXOX, you get two kinds of peaks. You get these early open peaks, and they are typically bound by OXOX and KLF. And you have these transient peaks. These are kind of the weird, interesting peaks. They contain low affinity OXOX motifs with AP1. And what's happening is, I think analysis indicates that AP1 is being dislodged from the enhancers, fibroblast enhancers, to these transient enhancers. And then these transient peaks eventually go away because the OXOX concentrations drop and occupancy at these binding sites go away. These ones stay open because they have high affinity binding sites of OXOX, so they stay open. And then ultimately, these late open sites, they require additional transcription factor motifs like ZIC. They come in and open up, and these late open peaks eventually transition into the iPSC state, right? So just to summarize the findings, I think what we show is during early reprogramming, the OSKM concentrations, transgene concentrations really spike. And then these OSK low affinity binding sites, they are made accessible by high concentration of OSK. They are defining these transient peaks, and then they get gradually released as a function of concentration dropping. And the decay rate of transients is directly related to the affinity of these motifs, right? So in addition to these weak affinity OXOX sites, the transient peaks also contain these AP1 motifs. And these AP1 motifs seem to be acting like sponges. They're entirely new sites, 10,000 new sites opened up. They're taking away AP1 from the fibroblast enhancers, turning them off. And so in some sense, our model has provided really nice cis and trans evidence for a potentially functional role for these early transient peaks. So they are kind of these strange peaks they appear to be essential to help reprogramming happen. There are additional experiments that our collaborators have done to kind of show this in an indirect way. You can create an AP1 dominant negative, right? And if you basically repress AP1 artificially early in reprogramming, those cells in fact reprogram faster. So the key point is you have to turn off the fibroblast program as quickly as possible while turning on the iPSC program in order to get actual functional iPSCs. Okay, so a really nice association between OXOX stoichiometry, somatic factor redistribution, and somatic identity suppression. And all this story is in fact told by a neural network and interpreted by a very talented graduate student. And as you might see, this sort of system is a general framework, I think, for understanding chromatin expression dynamics and really building a strong understanding of how all the components of gene regulation play together, TF stoichiometry, affinity, syntax, in various differentiation reprogramming systems. You could easily apply this to any of those systems. And with that, I'll stop. So again, thanks to Surag for really driving this analysis. When we started the project, we knew nothing about reprogramming, and we now know a little bit. Also, Lakshman and Akshay helped quite a bit. And then Mo is the mastermind behind all the experiments, absolutely gorgeous data from Kevin's lab. And then there's also been a strong collaboration with Helen Blau's lab, who in fact did some of the AP1 dominant negative experiments in a previous paper. And thanks to NIH for funding.
Questions? Okay, nearby.
Hi, Anshul. Thank you for a wonderful talk. I have sort of two questions, one pretty specific and the other one philosophical for the future. Did you look at the AP1 locus itself as to how are the chromatin dynamics? Basically, you need to repress the expression of transcription factor. And the other question is, sort of your data seems to suggest that what is really driving this induced pluripotency is sort of this low affinity binding due to an unusually high concentration of these factors, right? And I was wondering if you ever looked or planning to look to actual embryonic stem cells, right? To sort of solve this debate on the stem cell field of sort of how similar are your iPSCs, right? Because you're inducing the state with these low affinity factors that will not normally happen. And so if you compare embryonic stem cells to your generated iPSCs, whether you can trace what really iPSCs can do and how they look, and what are the things that you can never replicate. So excellent. I know the second one is a little bit of a...
Yeah, no, both are very good questions. So we have looked, in fact, at the AP1 locus, and I think what's really happening is AP1 self-regulates itself. And so I think there is a massive negative feedback loop. You start redirecting AP1. We don't see kind of some new repressors coming in. It might be a function that of chromatin accessibility not catching a lot of repression mechanisms, but in fact, we do see repressors in fibroblasts if we want to, like ZEB1. In the AP1 locus, what we're primarily seeing, I think, is self-regulation. So it's just sort of a rapid negative feedback effect. Your second question is really interesting too, and I think we haven't looked at it as much in this system, but we had previously published exciting work looking at iPSC-derived heart cells against actual in vivo fetal heart cells. And using these models, we can exactly dissect the differences and also figure out ways to actually improve the end states by figuring out regulation. So we haven't explicitly looked at iPSCs versus ESCs. For the purpose of this study, everything we see in the iPSCs correlates with what we see in actual embryonic stem cells. So these transient peaks, so forth, are not seen there. It's the same kind of behavior. But for systematic comparison between the two, it's certainly possible. We haven't done it within the scope of this work. We've done it elsewhere. But the models can tell you a lot of interesting things about those.
Thank you for the amazing talk. Did you look at why you get the new CTCF site and the nanog locus?
Yeah, we hope to have an entire vignette on that. We initially thought it was DNA methylation. It's not. It's actually unclear. So in fact, if you look carefully I had this plot of like the motif lexicon changing over time. And the gray box was CTCF, and it increases quite dramatically. So CTCF typically is considered to be a TF that has like approximately similar number of binding sites, but between fibroblasts and ESCs, there seems to be a dramatic increase in CTCF binding sites, and particularly in heterochromatin. So H3K9 trimethyl heterochromatin fibroblasts, typically a lot of new CTCF sites seem to be opening there. We don't know why or how they are opening up only at that time point. We thought it might be DNA methylation, but we couldn't find any evidence for that. So that's a great question. We have not solved that problem yet.
Do you get lower affinity sites?
It's not conclusive. We looked for that. It doesn't seem like it's affinity in that situation. It might be some syntax combinatorics. We need to look more into it, but it's nothing obvious. We looked at the affinity angle. It wasn't that, and it wasn't DNA methylation. So yeah, we don't know. Great question though.
The motif looked a bit different, but
yeah.
Okay. Yeah, so great talk.
Thanks.
My question goes a bit in the direction of the first one. So the reprogramming is an artificial direction. Mm-hmm. So you see all these mechanisms, but probably not evolved for that. So what part of what you see in these transient things is also potentially present in the forward and natural way? Is this bounding a mechanism that is actually selected for, etc.?
I think people have seen sponging in some other differentiation systems too. I don't remember exactly which one. So I don't think it's exclusive to this reprogramming. But how this happens in this reprogramming system, it's kind of difficult to, like you said, it's not evolutionarily conserved. There is no there's no pressure, evolutionary pressure to actually make this happen. I think it just happens to be that these low affinity OX-OX binding sites happen to be close to AP1 sites. It might explain why fibroblasts actually, amongst all the somatic cells, program the best in some sense with OSKM overexpression because of this kind of chance event just happening. We've been looking forward to trying this out in other reprogramming systems. Like you could reprogram PBMCs, to iPSCs and other somatic states and see how that works, right? Is this a universal mechanism for all reprogramming systems, or is it like this really kind of happen chance kind of thing for this system, right? But then the key point is that like this weird transient cell state that pops up with all these peaks, they're really unique to the system, and they have this really interesting property which, is not conserved in any way, but clearly is important for reprogramming.
Hi, Anshul. That was really cool. My lab does iPSCs to other cell types. This helped us to understand a lot. I have one question more on the experiment. When you do the overexpression of the 4 TFs, a lot of the times when you do differentiation or reprogramming, you're using an optimized sequence, like a human codon optimized sequence to do that reprogramming. When you're looking at the OSKM concentrations, are you looking at both the endogenous OSKM fluctuations and concentrations, or are you only looking at the transgenes? And if so, are there, do you envision a very controlled way of actually experimentally controlling the OSKM or any reprogramming concentrations for future experiments?
Amazing. That's my next slide. Did we speak before this? No, I'm just kidding. So what's really cool is what Surag found is that the Sendai constructs actually have a different isoform structure than the endogenous genes isoforms. So you can actually computationally deconvolve endogenous and exogenous. And in fact, in the paper, we have the two separate, the two together, and we can see really nice interplay between the exogenous initially expression and then, initially being highly expressed, and then they disappear completely, and then the endogenous takes over. So in fact, you can do this computationally. You don't need to necessarily look at, Thanks.
Hi. So you showed with the local interpretation using deep lift or deep shaft or integrated gradients, and also with the global interpretation with TF Modisco, you found the binding sites of known motifs. Did you find any new de novo sequence motifs that you have no idea what their function is or what binding site they are?
We did not find any novel TFs, but I think all the low affinity stuff that I showed you, those low affinity OX-OX sites, those are impossible to find without our model. Because they're just so non-canonical. If you scan them, you'll find them all over the place. And if you look for the canonical motif, you won't find it, right? So that's I think a really, really interesting. That's the novel aspect of what the model showed. And then, everything else seemed pretty canonical.
Okay, so and can TF Modisco tease them apart and show different?
Those sub-motifs actually. So TF Modisco is sort of multi-scale clustering, right, of motifs. So if you just look in higher resolution scales of clustering, you actually pick up all of these beautiful submotifs, and they perfectly correspond to kind of losing one domain, losing another domain, central in the PU SD having a complete mismatch, low affinity mismatch. So, and actually, I think we have evidence from another paper showing, I think it's an in vitro experiment showing that at really high concentrations of OCT4 and SOX2, in fact, that motif is found in vitro experiments too, the one without the POU HD domain.
Yeah, there's always the discussion of first motif and secondary motifs. So
yeah,
it could correlate.
Yeah.
Any other questions? Okay, I'm going to read the online question then. So one question is, how do we know that the binding site binds OCT SOX if the motif is non-canonical? I guess you partly answered that question, but...
Yeah, excellent question. I think, so one way would be ChIP-seq, and in fact, others have done bulk ChIP-seq experiments in this, and in the paper, we in fact do have nice evidence that there are actual binding occupancy happening through ChIP-seq, but I think the stronger evidence at the single cell or the cell state resolution is the footprinting. So we see really nice footprints, as I showed you, changing as a function of affinity and concentration. It is not experimental evidence, but it is certainly some orthogonal evidence that these low affinity binding sites are in fact occupied.
Any other questions? No more? Okay. So maybe I can ask one question. So as far as I understand, the model right now doesn't input the concentration of the TFs in any ways, right? But with your results, now does it make sense that we can biologically inform the model so that you can discover more motifs?
Absolutely.
Yeah.
So I think what we're showing here is just by using sequence models and transitioning across cell states, you get indirect evidence the model like learned some effect of the interaction between concentration and affinity. And so in fact, we saw this like a year and a half ago, and that's when Jacob actually started working on his models. He'll talk about it tomorrow in RegCis. It's a cis-trans model. It's a single model for all cells in a single cell data set. And it integrates trans information, TF concentrations with sequence to predict accessibility. That in fact does work very well, and there you can directly link up concentration to affinity and all the nice things.
Okay, great. Oh, there's one more question, I think. You forced me to exercise. That's good.
Sorry for the second question, but one big question for differentiation or reprogramming is identification of new transcription factors that can drive specific cell states. Right now, all the outputs of the model are a function of concentration, or you're using concentration of known factors that you're actually putting in. But is there a way, would you envision a way to say, hey, what about other factors I could have used for cell differentiation? How can you map the cell states back to those factors and understand their motif interactions and all their other interactions?
Yeah, so lots of ideas here. So just to be very clear, the model doesn't take in any inputs other than sequence and accessibility. It is true that the model is being driven by Oxox scale-up expression, which Yamanaka had found before. But let's play a game where you take transcription factors, you create Sendai vectors with all different combinations of transcription factors. You actually take advantage of the inefficiency of infection, and you just infect tons and tons of cells with all kinds of combinatorics of transcription factors, and you see what happens. You build a big single cell atlas, you study its dynamics, and then you find cells that are moving in the right direction, and gives you all kinds of cool candidates for differentiation reprogramming. So that could be done. I think there are interesting experiments Jason Bonrostro has done, and Will Greenleaf is doing, and others. But it could be a really nice way of like completely agnostically, using a screen of sorts and using a model to then tear apart exact mechanism to drive you in the right direction.